So let me introduce today's speaker. This is like a fourth speaker in our Colloquium series, Mike Hinczewski from the Case Western Reserve University. He received his PhD in 2005 from MIT. He was working with Neha Burker, who's a statistical mechanician, physicist. And after that, he actually spent some time as a postdoctoral researcher studying the biophysics. He has done a lot of interesting work. He was a postdoc with David Malai as well, who is my former advisor. And he actually joined the Case Western Reserve University in 2014 as a faculty member. And since then, he is working on also many interesting problems as well. So he has done evolution and also signal transduction, and kind of he's using this theoretical physical rule to understand the biophysical phenomena. So, okay, Mike, please.
Okay, so thanks, Changbang, for the kind introduction, and thank you for the organizers of the series. I saw that on the webpage there seems to be a lot of really kind of interesting talks. So hopefully this won't disappoint. So what I want to talk today is kind of, as Changbang mentioned, what my lab does is kind of use ideas from statistical physics to think about a variety of problems in biophysics. And what connects these various problems, whether you're talking about like single molecule level things like cell adhesion, or signal transduction, motor proteins, or to larger scales, applications like biosensing, is that at some level all these problems involve fundamentally stochastic processes. So whether it's the biochemical reactions involved in cell adhesion or the dynamics involved in motor proteins, there's some ideas here that make it a natural fit for statistical physics as a description of these systems. And that's kind of what drew me into biophysics, and that's what my group continues to do. And in the last few years, we've actually been looking kind of at larger scales, because we realized, for example, some of my postdoctoral training has been, as Changbang mentioned, with Dave Thirumulai, looking at things like protein folding and ideas of like diffusion on free energy landscapes. It turns out that there are very close analogies to when you look at things like evolution, like fitness landscapes and diffusion on fitness landscapes. So it's been kind of a natural transition for our group to think about dynamics and kind of larger scales, and also look at evolution as well. Now, the question that we kind of have become fascinated by in a variety of these systems recently is the idea of control. So if you have this fundamentally stochastic process in biology, can you make some kind of external intervention to allow to do something interesting, to control the system from the outside, maybe to shift the probability distribution from one form to another over some kind of finite time? And the story that I'm going to tell today is one aspect of this kind of control problem, where we're trying to apply these ideas of control in an evolutionary setting. So this is based on a paper that just came out a few months ago. And it's a collaboration with a variety of different groups, Effa Ilker at the Institute Curie, Sebastian Defner, who's a kind of theoretical quantum physicist at University of Maryland, Baltimore County, and Jacob Scott, who's kind of a theoretical oncologist at the Cleveland Clinic. And what started this whole process of thinking about evolutionary control for us is actually an inspiration from a very, very different area of physics. So that's this idea of quantum adiabatic computation. As, for example, in these kind of commercially available D-wave systems. And the way the quantum adiabatic computation works is also a kind of problem of control. So there you have a quantum system. So you can model it as essentially a set of spins interacting through various potentials. And at each spin, you have the freedom to apply local magnetic fields. Right? And then you can create a fairly complicated system with these degrees of freedom. And the idea of adiabatic quantum computation is that for a given set of local magnetic fields, if you can find the ground state, that in some sense encodes the answer to your computational problem. So the idea is like, can you somehow build that ground state? Once you have that, you can read off the solution. And the way it typically works is that you start with a much simpler system. So for example, if I put all the local magnetic fields to point in one direction, the ground state is going to be all the spins pointed up. And then if I manipulate this system, so if I now slowly change my local magnetic fields to their final configuration, We know that if we do this slowly enough, we're guaranteed by something called the quantum adiabatic theorem to always remain in the ground state of our system as we make this perturbation. So at the very end of the time, as we make these very, very slow changes in magnetic fields, we eventually end up in that final ground state that is the answer to our problem, and then we can read it off and we've solved this particular computation. The problem with this idea is that for a practical computer, slowly changing things is not a very useful thing, right? We can't spend an infinite amount of time to read out the answer for a computational problem. So people have, to make this actually work, people have devised all sorts of ways of trying to speed this up. And the idea here is to avoid the problem that you get if you just naively quickly change local magnetic fields, because then there's always a chance that you will end up either in an excited state or some superposition of excited and ground states. And if you do that, you're not guaranteed then to get the correct answer. So how do you do this? Well, in the early 2000s, a variety of groups, including Demiplak and Rice and Michael Berry, devised a really simple strategy, which is you take your original Hamiltonian, you add a certain perturbation to it in such a way that you're guaranteed to always to remain in the ground state of your original Hamiltonian. And this is this general field of kind of speeding up adiabatic evolution is called shortcuts to adiabaticity. And this is one particular shortcut, which is known as the counterdiabatic driving. The way this works concretely is, let's say you have some time varying Hamiltonian, and that's our original Hamiltonian of interest that encodes, whose ground state at the end at time TF encodes our answer. And let's say its ground state at any given time is, we'll call that phi 0 of t. Then what we say is, well, we're going to devise a new Hamiltonian, which is that original Hamiltonian plus a counterdiabatic perturbation, so HCD, that obeys a Schrodinger equation like this. And we're going to choose that HCD perturbation in precisely such a way that the solution to this new Schrodinger equation is exactly the old ground state of the original system at all times. And this is the expression from for example, Berry's paper. And what's really nice is that you can have this analytical closed form expression for exactly what kind of perturbation you need to guarantee this. And it's entirely expressed in terms of the eigenstates of your original Hamiltonian. So if you know your original Hamiltonian, you know the eigenspectrum, you can get this expression. Now, even though this is kind of a beautifully elegant analytical solution, there's lots of practical problems, because in practice, you may not be able to actually implement such a perturbation experimentally in your system, right? So there's still a lot of work to be done to make this practical, but as a conceptual kind of proof of concept, this is a very nice idea. So what's nice also about this is that we know that quantum systems are in their way that they're described, we have a wave function that's evolving in time, have very close analogies to classical stochastic systems, where you have, for example, a probability distribution evolving in time. So you can actually try to draw mathematical analogies between these quantum systems and the classical ones. And that people have begun doing that in the last few years. One very elegant example of this is an experimental setup where they used optical tweezers. And the way this works is you have a bead in a laser trap, and the potential felt by the bead is, roughly speaking, a harmonic potential. And you can control the spring constant of the harmonic potential by basically changing the laser power, right? So you can make it stronger, you can make K steeper, you can tune down laser power, you can make the spring constant smaller. And this way you can have some kind of external control on the system. And in this case, if you look at the fluctuations of that bead, they're going to be described by some probability distribution. Let's say we're only interested in the one-dimensional, displacement, so x. So the probability of being at a certain position x at time t. And that probability distribution obeys here the classical analog of what we had before with the Schrodinger equation, and this is a Fokker-Planck equation. So it depends on, there's a term which is the drift velocity term in this equation, which depends on the shape of the potential. So it's encoding the force felt by the bead. And there's another term which encodes the fact that you have thermal fluctuations, and that's just the diffusion coefficient term, like that. So you have this equation, so you know how this probability evolves in time. And let's say, for simplicity, you just kept the trap at a constant strength. So K was constant. Then we know what happens. Statistical physics tells us if we wait long enough, the system will eventually equilibrate, and you'll get a distribution which just looks like the Boltzmann distribution, right? Which is shown here. I will write it as P with superscript EQ. And it depends, of course, on the spring constant K of the trap. Now, what happens though if you were to change that spring constant over time in a protocol? So let's say, for example, you started with a small spring constant at early times, and you wanted to raise it to a large spring constant over some finite time interval. Well, at early times, let's say your system has already equilibrated. So here's your potential, let's say at time point TA, which is at right over here. And the red here is the actual probability distribution. The blue dashed is the equilibrium corresponding to the spring constant K0 initially. Now, as you begin to increase the laser power, and so the spring constant increases, your instantaneous equilibrium distribution, so basically the PEQ at that particular value of the K corresponding to that particular time, is going to just follow the potential. So that's going to immediately begin to become, the distribution is going to become narrower, but it's going to take some time for the actual distribution to catch up, because you're basically driving the system out of equilibrium. And so there's going to be a lag between your actual distribution and your instantaneous one. Now, once you stop changing, once you get to point C and you wait long enough, of course, it'll catch up and you'll go back to equilibrium. But in between, you've essentially went out of equilibrium. And this is equivalent in the classical sense to the problem in quantum mechanics where if you do a finite time protocol, you're driven out of the ground state. So the instantaneous equilibrium is essentially the analog of the ground state in the quantum case. If you do this, of course, infinitely slowly, the red and the dashed curves will always overlap and there'll be no problem. So the idea is, can you then fix this? And this was what was done by the group of Martinez et al. This was a Nature Physics paper from a few years ago. And you can easily calculate this, what kind of potential you need to add in order to guarantee that you will remain at the instantaneous equilibrium of your original protocol. And it just turns out that it's a small perturbation to your spring constant, shown here on the right. And with that perturbation, you can guarantee that you're always, in instantaneous equilibrium. And the other thing to note is that the perturbation depends on the rate of change of the spring constant. So naturally, as you make that smaller, so if you go to the adiabatic limit where you're infinitesimally slowly changing everything, you will end up, that perturbation just goes to zero. So you recover the adiabatic limit. But if you're doing it very, very fast, that perturbation has to be very, large in order to keep you basically at the, is there a question?
Yeah, I have a question.
Okay, sure.
Here, if it's a real optical trap, the time scale of this bead thermal motion is, I guess, microseconds or less. So the time interval between T0 and T1, should it be even shorter, like nanoseconds or close to femtoseconds?
I think if the bead is large enough, and I have to double check this, the relaxation time, I think, is on the order of milliseconds. But I have to I don't recall now from this particular experiment how quickly their protocol was. But I think I think roughly speaking, it's on the order of milliseconds. So yeah, you're right that you do have to, in this particular experimental case, drive it faster than the equilibration time. Because if you're driving it slower, then of course it's always going to instantaneously equilibrate and you won't see this deviation. But since they were able to experimentally see this, so I think the time scale, reaching the time scales necessary was not an issue.
Okay, thanks.
Sure. Okay, so that's the case for this kind of nudging, this counterdiabatic nudging in the optical tweezer. And what we wanted to do was essentially see if we can take these ideas and now apply them to what, in our opinion, is a really much more interesting and complicated problem, which is evolution. And we were inspired by these experiments that I just described. We were also inspired by theoretical work by Ayodi Patra and Chris Jarzynski, which looked at how you might define these types of problems in general in a classical sense. And we thought, okay, hey, can we do this for evolution? And what's the motivation here? So why would we want to control an evolutionary system? Well, this comes a lot from our collaboration with the group of Jacob Scott in the Cleveland Clinic. And they're in particular, they're interested, it's an oncology group, so they're interested in the issue of drug resistance for tumors. But there's also, it's very, very closely related to drug resistance for bacteria. And oftentimes bacteria turn out to be an easier system to work with experimentally. So actually they both work on, tumor evolution and bacterial evolution. And bacterial drug resistance is something that doesn't need any real introduction. Everybody is familiar with it. This is an example of a kind of a nice kind of visualization of it, where it's what's known as a bacterial lawn type experiment, where you put antibiotics on a Petri dish and you let the bacteria grow. And you can see that in certain cases, the bacteria don't want to get close to the antibiotic because the antibiotic is diffusing out in the surface and killing the bacteria. But in this particular case, this bacterial variant has resistance, for example, to the antibiotic on the right, or here on the left, where I can go really close up to it because essentially it's unperturbed by the presence of the drug. So we know that bacteria, because of their propensity to mutate, can develop resistance to a variety of drugs. And it's kind of an ever continuing war, which we're seemingly unlikely to win in the end run. Because as fast as we can innovate new treatments, bacteria can evolve to evade them. And unfortunately, the same is true also for cancer evolution. So this is a slide which I borrowed from my collaborator, where this was a patient that came in with a type of a very bad melanoma, and he was given one of the best, kind of the newest generation of drugs designed specifically for that melanoma, and it seemed to work wonders. So after 15 weeks, the cancer seemed to be entirely in remission. But unfortunately, some of the cancer cells were left. They were able to mutate, and the mutations conferred resistance against the drug. So several weeks after that, the cancer returned and the patient died. And this is, again, just like in the bacterial resistance, it happens all the time. It's just that it doesn't happen on the order of days or weeks. It may take many weeks or months. I mean, the timescales for tumor evolution are longer, but it still eventually happens. And so there's this need to basically come up with some kind of clever approaches that basically take into account the fact that evolution is always going to happen. We're never going to get rid of evolution in the system, but can we somehow develop medicine to compensate for the fact that evolution has to occur? So the idea here is, this is one of the strategies in what's known as evolutionary medicine, is let's say that you have several different drugs at your disposal. And it turns out that there is a phenomena where certain variants may be extremely fit or relatively fit under one drug and be resistant to it, while at the same time being extremely susceptible to a second drug. This is known as collateral sensitivity, and it's actually been experimentally demonstrated for a variety of different drug pairs. So you can have quite different behaviors of the same variant under two different drugs. And can we take advantage of this? So for example, let's say we start with a population of different genetic variants. And here, just for simplicity, I have 3, but in reality, there's often many more. I denoted those variants A, B, and C. The size of the circles represents the overall relative population in this given infection. And the color coding here represents the fitness. So if it's kind of pale and whitish, it's low fitness, and the bluer it is, the higher the fitness. So let's say you begin to apply, a first drug. So you increase you give the drug, you wait over time, and because of mutations, you'll have some of the population of A cells mutate into type C cells, which are highly resistant. And those resistant cells, and this is a classical, drug resistant case, will eventually come to dominate the population, right? But the idea here, that normally is a bad thing, but the idea here is that you've done this in a strategic way, that you wanted the cells to basically go to entirely type C, because you have a second drug, which type C is extremely susceptible to. So under the application of that second drug, now C has extremely low fitness, and you can eradicate it before it has a chance to escape into any kind of resistant strains. And so this is kind of the ultimate goal of evolutionary medicine, or this type of evolutionary medicine, is to design rational drug protocols to exploit the evolution in order to basically combat the tumors. And the interesting idea here is also that you have to, the choice of which drug to apply and in which order becomes extremely important, right? If we switch the order here, it would not work, right? So we have to do this in a kind of rational way. But in order to do that, of course, we have to understand the fitnesses of all these variants under the drug. So ultimately speaking, control has kind of 2 issues, which I want to mention kind of really talk in detail about one of them, but I want to mention both of them. So the first issue is, like I just referred, is essentially you need to know the fitnesses. And this can become a really challenging combinatorial problem. It's really difficult because you have to basically do these individual kind of growth rate experiments on many different mutants. So some of the largest data sets available up to now are on the order of like 16 mutants, and you look at 15 different drugs and maybe one dosage level of each drug, right? And that in itself is a huge amount of work, and you get diagrams like this representing, you know, the 16 mutants and the relative fitness under different types of drugs. Yeah, so that's one thing. So getting these kind of fitness landscapes for the drugs is kind of a prerequisite to doing control, and is a big challenge. That's something that our group is considering. But I'm not going to talk about it for this talk, but it's one of the things that we're working on, is what techniques can we use to infer the fitness landscapes. And ideally, ultimately speaking, what we want to do is kind of in an iterative way, both infer and control at the same time. So we make a perturbation to the system. Let's say we don't know about the fitness landscape. We use the response to the perturbation to make a guess about the landscape, do another perturbation based on the guess, and kind of iteratively do this until we, you know, as we improve our model of the landscape, we get, you know, better and better control along the road. So that's kind of the holy grail eventually, is to be able to do this for landscapes where we don't have that information. But we're gonna, yeah.
I have one question. So can you make it clear that the this evolutionary landscape, and then your some comment on the optical tweezer, the, you know, distribution of particle actually changing from one Gaussian to another Gaussian by perturbing the system by changing the spring constant, right? So how that is related with this story? I mean, I see that
I will show that in a second. So it turns out it'll also be through a Fokker-Planck equation. And it won't exactly be mathematically analogous to changing the spring constant, but we also will be, at least in the simple case, you know, effectively changing the landscape. And in the more general case, we'll be changing the drift velocity. And I'll go into the mathematical details in just a couple of slides. Okay. So I'll try to make that connection clear. Yeah, but that's a good question. But I will try to make that clear.
There's one question. What do the arrows in the diagram mean?
So I believe in this particular case, they just refer to transitions that go up in fitness. So in general, those arrows are things where you can have mutations, and the direction of the arrows just represents that that mutation takes you up in fitness. So, you know, you're more likely to see that, you know, in these, you know, that's essentially what those arrows mean. You know? Okay, so the other problem that you would encounter just if thinking about controlling these things is that, imagine this kind of 2-drug therapy, right? So this idea is that you would apply one drug for a certain amount of time, you'd wait for the system to get to the final destination, kind of equilibrate at the distribution that you're interested in, which is mostly C, and then you would apply the second drug. And you might imagine that this could be extended to like a multi-drug, you know, protocol over time. But just sitting around waiting for that equilibration to happen is again not a very practical thing, because that might take days, it might take weeks. It depends on the mutation rates, you know, and other parameters of the system. So you can't take this to a clinical setting and tell the patient, well, you're just gonna have to wait around for your infection to equilibrate in an evolutionary sense, and then we'll treat you, right? So is there a way of basically speeding up this equilibration? And more generally, getting from whatever distribution we start with to our final target distribution, you know, in a finite time, right? That's the ultimate control problem. That's kind of what we're gonna, you know, the main focus of the talk is, like, if we have some kind of, you know, therapy, which may be the first stage of this, you know, collateral sensitivity type 2 drug protocol, can we speed up each stage to get to the target in a finite time? That's the basic question. Okay, so to do this, we're going to have to set up some kind of mathematical model for evolving cells. And how that works is, well, let's say you have a cellular population and you have several different types of genetic variants. So we'll call the number of variants M, capital M. In this case, it's 3. We're always going to use the convention that the mth one, so in this case, the type 3 is our, you know, reference type. We'll call it the wild type. And we'll think of the other types as essentially mutations off that reference. But in some sense, the reference type is just a convention. So what happens to this population? Well, the cells can give birth. In this case, I'm showing just simple binary division, but the rate at which that occurs might be related to the fitness of each type. And so they might have different fitnesses in your particular environment, right? So if your environment is full of drug, certain cells are going to be resistant, certain cells are going to be susceptible. That might affect their rates of cell birth or equivalently rates of cell death. You might have mutations, so different types can change upon division. So you might end up switching different types, and you also might have cell death, which can also be dependent on your environment. Ultimately speaking, we're going to be talking about kind of this fitness in terms of fitness relative to the wild type. So if we have our fitness divided by our type I divided by the wild type fitness -1, we're going to call that the selection coefficient. And if S of a particular type is greater than 0, that means it's fitter than the wild type. If it's smaller than 0, it's less fit than wild type. And what our external control is going to be is we're going to be able to essentially do something to perturb this value of S. And the idea here is that this S might depend on some kind of time varying external parameter, which we'll call lambda. And I'll write lambda as a scalar, though in principle, it could be a vector of many different types, a multidimensional vector. We can have many different control knobs. And the simplest scenario would be that lambda would be the dosage of some drug. And it's going to impact the growth rates of the mutant and wild types generically in a different way. And so you'll have some function, each S will be some function of this lambda. And when you now have this, if you thought of the S's in some sense as defining a fitness landscape, now that this landscape is changing over time, it's known in the literature as a fitness seascape. So you essentially have this, externally varying, landscape, which is actually a kind of a nice terminology, which I wish the protein folding people would adopt. But the first time I heard it was in the context of evolution. So you go from a landscape which is static to a seascape, time dependent. And like I said, the lambda here could include the multi-dimensional. It could include, you can have multiple simultaneous, drugs applied. You can change temperature as well, nutrients, osmotic pressure, etc. So there's no end of external factors that in some sense might influence the fitness of the system. Just as a concrete example, which we'll return to numerically later on, this is actually one of the nicer systems in that they've done a kind of complete mapping of the seascape of how these fitnesses depend. This is for a system where they're focused on a gene called DHFR, which is naturally expressed in the malarial parasite, and there are different variants of it which have different degrees of resistance to a typical anti-malarial drugs. In this case, the drug is pyrimethamine. And what they've done to make it easier to study is they've taken this gene, put it into yeast strains, and they've looked at all possible mutations of this gene. So they know that in malaria in the wild, there are 4 possible sites of mutation, and if it's the original is you call it 0. If it's mutated, you call it 1. So they're essentially combinatorically 16 possible different mutants, right? So at each site, you can either have a 0 or 1, depending on if it's mutated. And they look at all 16 different types of this gene, and they study in this transgenic yeast system what the growth rate is as a concentration of drug. And so this is the growth rate versus drug concentration for each of these different types. And you can see, for example, here that the type which is labeled 1110 has the largest growth rate, so it would dominate the population at low concentrations of drug, and then but it changes, right? So there's some complicated interplay where the rank ordering of which is the best and which is the worst changes as a function of drug. And this is an example of what I would call this a seascape data for this evolutionary system. Now, okay, so let's say you have all this data. You still need, in some sense, to be able to describe it mathematically. And there, what we use is something called the Wright-Fisher model. And this is in some ways like the Ising model of evolutionary biology, because you can take any kind of realistic model of how individual populations grow and divide and map it onto a Wright-Fisher model. It turns out to be quite useful in this sense, even though the model itself seems a little bit stupid. It seems almost like a spherical cow model of evolutionary biology. Lots more other realistic models can be mapped onto it, and so it's almost the default model that people use to study evolutionary systems. So to kind of describe it very briefly, I'm just going to imagine a system with two gene variants, A and B. And in this model, the simplest version is that you have a constant population size that you can relax that and easily generalize that to varying populations. And so let's say that the type B has a fraction x and type A has a fraction 1 minus x. And the way the model is defined is we look at generation to generation and we ask what is going to be the distribution of A and B in the next generation. How that occurs realistically is maybe complicated, but in this model, it's done in a almost a little bit crazy, but it turns out mathematically not, still meaningful way. So if you had no mutation and no selection, so what we call a vanilla Wright-Fisher model, the way the next generation is chosen is basically you choose your parents randomly from the previous generation. So you can draw lines to the previous generation and then just copy over the type from the previous generation, right? And if you do that and you look at the fractional change in the B fraction, so delta x, and you do this, of course, over an ensemble, so you repeat this many times, and so you can calculate ensemble averages of the delta x, it turns out that the average of delta x doing it just like this would be 0, but the variance in delta x would go something like this, like x times 1 minus x divided by n, where n is the size of the population. And this captures something which in evolution, which is known as genetic drift. So it's essentially because your next generation is always in some ways a sampling of your previous one, because your previous generation has a finite size, you're not guaranteed to capture, make a perfect copy of it, right? You might have a type in your previous generation was extremely fit, but runs off a cliff and dies, and you lose it, right? So there are lots of things that can happen within the course of a generation, and that type may not survive until the next generation, even if it is super fit. So there's some kind of random sampling always involved, going from one generation to the next. And of course, if you go to an infinite population, this goes to 0. But if you have a finite n, this is a non-zero quantity. Okay, so what do you do? How do we add natural selection to this? So here, then, what we do is, let's say so this idea here was that you basically choose your parents totally randomly, independent of what type they are. But let's say you have a slightly larger chance of choosing type B than A, and that chance is exactly 1 plus s times. So that essentially means your selection coefficient, the ratio of the fitness of B and A is 1 plus s. In that case, you now have a bias. Your average delta x has this non-zero term, so it's proportional to s. To 0th order, you still have the same variance, but now you see that if, for example, if s is positive, so B is slightly fitter than A, you'll have this tendency to increase on average the fractions of B's because of natural selection. And the final element that you put in there is you allow the types to mutate. So for example, A can mutate to B with some rate, B can mutate to A with some other rate. And when you do that, you now get two other terms into your average delta x. And now you have what we consider kind of a very rough but complete model of an evolutionary system. And any realistic system, if we look, for example, in the paper at an actual realistic model of individual cells growing and dividing, you can more or less map it onto these two terms where you get an effective mu's, you get effective s's, etc. when you do this mapping. But as a mathematical description, this actually works really nicely. All right, so what do you do with this? Well,
Mike,
yeah.
Can I ask a question?
Sure.
So is this a discrete time model?
Yes, in a way, it's a discrete time model in that you're looking from generation to generation, right? So you have your time steps are Wright-Fisher generations. What we're going to do next is actually map it onto a Fokker-Planck model, and that model becomes a continuous time model, but it holds when you look at timescales which are like hundreds of thousands of generations long, right? So if you zoom out onto really large timescales, you can then treat it as if it was continuous time. But in the original formulation, the Wright-Fisher model is discrete. And there are variants of this which you can look at more microscopic timescales, but this is the kind of canonical model that people use.
And the last term in the delta x average value, that is s times x times parenthesis 1 minus x. That looks exactly like a logistic equation type.
Yeah, it does. And you can kind of rationalize it in the following way. So let's say we're just looking at selection, so we don't have any mutation here. If we start entirely in a system of all A's, so our X is going to be 0, and in that case, there's no in the absence of mutation, there's no way of getting a B in the system, and so you're not going to get any delta X once you're at X equals 0. And similarly, if you're entirely in B, right, so your X is equal to 1, that term will also be 0, because there's no way, you won't have any benefit of selection if all your variants are exactly the same type. So that's essentially what that term captures, that X times 1 minus X.
So even though this may not be exactly a logistic equation, but the range of the S value might cause some chaotic behavior if it is too big.
I don't, yeah, that is a possibility. I don't know if, I haven't personally looked at it because the typical values of S's that people consider are usually much, much smaller than 1.
I see.
But yeah, so I haven't looked at what happens in the really large S regime. But there's a possibility, yeah. It might have a very weird, somewhat weird behaviors. But in a realistic sense, oftentimes, things that are extremely, let me put it, so the way people typically in evolution look at it is that the scale that matters for S is about 1 over n. So if S is much, much greater than 1 over n, that's considered to be really strong selection. And if S is much smaller than 1 over n, that's considered weak selection. And since n is typically a large number for evolving populations, the scales of S's that matter are usually quite tiny.
Okay.
Okay, so how would such a system behave? Let's say we kept S constant. So B has a slight advantage. And let's say the mutation rate back and forth is usually also typically a small value. We set n to be 10 to the 5. Let's say we start with a system that's entirely A, made of A's. So our X equals 0. We have some mutation here, so a mutant of B will appear. And since B is fitter than A by a little bit, it will actually be have a competitive advantage. And this is of course one realization. Of course, it couldn't just die out. But let's say it doesn't die out, then that mutant can come and eventually reach some kind of equilibrium, and that equilibrium is described by a probability distribution of the X's. And this is where we get to the kind of analogy to our optical tweezer system, because it turns out that these dynamics can be described by a very, very similar Fokker-Planck equation. Now, the velocity term here in the Fokker-Planck equation turns out to be just equal to the ensemble average of this delta X. So it's given by this. And our diffusion term turns out to be related to this genetic drift, and it's equal to 1/2 the variance of delta X. So it's equal to that. And this is something that's, it's been known quite a long time in evolutionary biology. So Kimura, who was one of the founding fathers of this entire field, kind of mathematical population genetics, this is from a 1955 paper of his. And it's a really kind of beautiful way of describing dynamics, because now you can connect it to a lot of things that we do in statistical physics. Later on, this is of course for a one-dimensional system. I'll later show you how it works in higher dimensions. Now, it turns out for 1D, there's a really kind of neat mapping onto kind of statistical physics ideas, which is that you can write the velocity term as minus d times essentially the derivative or the gradient of a potential. And if you work it out, you can work out essentially an effective potential for the system, and your equilibrium distribution, the one that you will eventually get to at long times if you don't change the parameters of the system, is just given by in a Boltzmann form with this effective potential. And so you can imagine, for example, that effective potential looks, if S equals 0, so you have no bias towards A or B, it's just a symmetric well, and you get a distribution which is just centered around 1/2. And if you make S a little bit negative, so you give some preference to A, it's kind of like tilting the well, and you move the potential over to smaller values. So you get more A, less B in the system. And if you make S positive, you tilt in the other direction, so you get more B and less A in the system. So it looks very much like, just this kind of a standard statistical mechanics system that we're familiar with from other contexts, but it does have this position-dependent diffusivity, so it makes it a little bit more complicated. what would happen if I decided now to make a time-dependent S, right? So the analogy of now changing my, in this case, the U here for this one-dimensional system, I'm now, changing that over time. I'm not actually necessarily making it, tighter or spreading it out, but I am tilting it, right? So let's imagine I changed S from a negative value favoring A to a positive value favoring B. And we can, in this kind of analogy, think of this as maybe a drug that, as you increase the dosage, B gets favored over A because B is more resistant to the drug than A is. So what I'm going to show in the dashed lines is the instantaneous equilibrium. So that basically this P equilibrium at the particular value of S corresponding to the protocol at any given time. And then the actual Fokker-Planck solution is going to be shown in blue. And so what you'll see is as I do this thing, the instantaneous one changes just along with the protocol. And the actual P, it takes a long time for it to actually equilibrate because mutation is slow, and that's the kind of sets the timescales for the equilibration timescales in these systems. So here it is again. So you see this kind of generic lag that that I mentioned also in the optical tweezer example. All right, so what we want to do eventually, of course, is get rid of that lag and to develop a counterdiabatic protocol. But we don't want to necessarily do this just for a one-dimensional system because these one-dimensional systems correspond to two genotypes, right? So it's always, the dimension is one less than the number of genotypes in your system. And in real settings, you might have many, many different genotypes that you work with. So I'm briefly going to mention just generalizing this to greater than two genotypes. So there, it actually works out quite nicely. You define an m-1 dimensional vector where the ith component of that vector is just a fraction of the ith mutant in the population. And you don't need to specify the last one because you know that the last one, for example, if there are 16, you know that the fraction of the 16th one is just 1 minus the sum of the fractions of the previous ones. So it's, you only have to worry about an m-1 dimensional space in terms of the dynamics. And you get a multidimensional Fokker-Planck, so it's a generalization of that 1D, system that I showed before. I'll mention briefly the various terms, but we can think of this generically as being some operator, a differential operator acting on our probability, telling us how that probability evolves in time. And that operator, the velocity term in particular, depends on our protocol. And how does that work? Well, the velocity term will have a contribution from mutations. So this M is just essentially a matrix denoting which types can mutate into each other. So it's just generalization of the mutation term that we had. And then we have a term which depends on selection. Before it was S times X times 1 minus X, but now in general it's a matrix G times the S vector. Right now you have a vector of S's because each type has a certain selection coefficient relative to the wild type. And the diffusion coefficient, is now a matrix and given by this small g matrix, which is defined here, right? So this is the generalization now to many dimensions of that one-dimensional example. And the important thing to just keep in mind is that this is the part shown here in red that depends on your protocol, right? Because as I change my drug dosage, for example, I'll be changing my S values and hence influencing this term. And that's where the kind of external control comes in the problem. And you can imagine in other cases, doing this, maybe if you have some kind of like ability to change the rate of mutation, stuff like that, there may be other ways of doing control, but this is by far the easiest way. It's just basically changing, the selection coefficient as a function of some external variable. All right, so what's the problem that we want to set up? So we're going to choose some control protocol, lambda of t, between initial and final time. That lambda of t is going to define a sequence of instantaneous equilibrium distributions. So basically P equilibrium at each value of lambda along our protocol. Each of these distributions are basically right eigenvectors of our differential operator, our Fokker-Planck operator, right? Because if that operator acts on P equilibrium, it should give 0, the instantaneous equilibrium. And the control problem is that we demand that our probability equals our instantaneous equilibrium probability at all times during this protocol. And that's what we want. And we know that the same challenge that we just saw numerically a couple of slides ago, if we just directly implement our original protocol, it will not generically work because the protocol will drive our system out of equilibrium, and our P will lag behind the equilibrium. So in a multidimensional sense, you can visualize this like this. For example, this is in the case of 3 genotypes. So let's say you have some control protocol, some drug dosage. You're changing from some initial to final time. That maps out a path in some vector space of your selection coefficient, right? This is a 2-dimensional space because we have 3 genotypes. And for each value of that S, there's going to be a corresponding instantaneous equilibrium. I wrote it as rho here, but it's the same thing as P equilibrium. And, so as we change our lambda, we get different target distributions. And just here, this is going to be important later on. These are the mean values associated with each instantaneous equilibrium. So the mean of this distribution, I'm going to write as X bar, corresponding to that individual value of drug dosage. And we know that if we were just to implement this original control protocol, generically speaking, the actual distributions that we get at, let's say, 3 different times, an initial, a middle, and a final time, they're going to generically lag behind, our instantaneous ones. So we're not going to be, we want to get rid of that lag. So what do? So the setup then is to kind of like in the original quantum case, we want to go to a new Hamiltonian, but in this case, it's a new Fokker-Planck operator. We'll call it lambda tilde. And we set it up such that the equation it satisfies, by construction, the solution is our equilibrium, P equilibrium. So then we know that if we can implement this lambda tilde, we're always going to be able to, to achieve the counterdiabatic control. And we also we want to have the adiabatic limit work. So we know that if our protocol becomes, changes infinitely slowly, we want to return back to our original Fokker-Planck operator as a consistency, right? So those are the 2, kind of conditions that we would like to implement. And it turns out that you can actually solve this in this evolutionary system exactly. And to do that, you need to actually implement a selection coefficient, a new selection coefficient, which now not only depends on the protocol and the speed of change, but also on the actual fraction. And this is known in evolutionary terms as a frequency-dependent selection coefficient. So what does that mean in practice? That means you have to, your fitness if you're in a population depends on how many other variants there are. Now, this is actually not something that's typically seen when we change drug dosages, and I'll mention that practically. So we'll have to do some kind of approximation because we don't, it's not easy for us to implement this in a laboratory setting. But in certain contexts, for example, ecological systems, which have which are mathematically quite similar to these evolving systems, you can get these kind of frequency-dependent selections. Because, for example, if you have 2 competing species, one species might create a toxin that interferes with the other species, and hence, if you're the victim species, your fitness is going to depend on what proportion of the other species exists. So it'll depend on the relative proportions of the 2 competitors. So in ecology, this occurs in kind of bacterial drug resistance. It's a little bit harder, but let's ignore the practicalities for now and just think of it as a math problem. So how would we find that S tilde? We can set it up so we can define a probability current, which is a function of that S tilde. And if we could find the probability current, we can invert this, actually, it's invertible, and figure out what S tilde is. Because everything else we know what P equilibrium is, we know what G is, we know what S is. So if we knew what J tilde was, we can invert it to find S tilde. And in this J tilde form, this new Fokker-Planck equation takes the form of a probability conservation equation like this. And it turns out we can invert this and get a form for J tilde, which looks like this. It's a little bit nasty, but it's an exact solution. And it turns out to have kind of some interesting features. So this X hat here is just the unit vector along the ith axis. This integral here is only carried out over one of the components, and there's a sum. So we look at component by component. So this is actually a sum of several different integrals. And the final thing is that there are 2 quantities, this Wi, which are just scalar numbers that have to add up to 1, and this function f here, which has to be divergence free. And the final condition that needs to be satisfied is that near the boundaries of your probability simplex, where this is defined, you need to have the normal, of the probability current, dotted into the normal equals 0. So you can't have probability flowing out of the boundaries. Despite the complexity of this, it turns out that generically speaking, for m greater than 2, there are actually infinitely many functions f and vectors w that satisfy these conditions for a given series of distributions. So it turns out that there are actually generically infinitely many possible CD solutions. And then the only question remains is, okay, mathematically this is fine, this is wonderful, but out of this infinite family, can you find ones that you can actually implement in real life? And this is, of course, the major challenge in general for these counterdiabatic methods, even for the quantum ones, is can you practically do that? It turns out that in the scenarios that we think are most relevant to our evolution, to our kind of laboratory systems that we're eventually interested in testing this out, the populations typically are large and the mutation rates are typically fast in these systems. And in that regime, it turns out that instead of using a fully x-dependent S tilde, there's an approximation you can make that essentially just becomes dependent on the protocol and the rate of the protocol changing. And in that case, rather than getting infinitely many solutions, the solutions converge on a unique value, a unique form, which looks like this. And what's really nice about this is that it's essentially a closed form analytical solution for what you need to do to the system, like what perturbation you need for the selection coefficient in order to achieve this driving. And what it depends on is not even the whole probability distribution, but just the average. So that path that we described here, that X bar path, which I showed here, if we know that, so we know the sequence of distributions and we know the sequence of average fractions, once we know that, we can write down what the perturbation is that achieves CD driving in this case.
Mike,
now, if this approximation doesn't, yeah.
Can I ask a question?
Yeah, sure.
So here summations are up to m-1, while there are m
Yes.
genotypes. So is there any kind of constraint or normalization condition?
Yeah, so it's just the way that the S's are defined. So because the S's are defined relative to the wild type, you can think of SM as being 0, because yeah, it's just the kind of, we define everything relative to one wild type reference, right? And that's and it just makes it easier to do that, because then you work in a dimensional space which is one less than you otherwise would have to. So you factored in the constraint that all the X's have to add up to 1 in effect by doing that. Yeah, and in particular this X bar M here, the average of the wild type you find by just taking 1 minus the sum of the others. All right. So once you do this, you can now have this nice closed form solution, and then you can ask, can your drug dosage, for example, achieve a perturbation to do this? And that's another question. And in the cases that we explored, we could get a fairly decent approximation, but of course, generically speaking, we may need not just one drug, but maybe multiple drugs, multiple tuning knobs to implement this term. But just just let me show you some simple cases where it works out nicely. So this is our one-dimensional case, meaning n equals 2 here, where before we saw that lag. Now with this new S tilde, which I called SCD here in this graph, this is our perturbed one. And what you can see is that now the actual Fokker-Planck solution, which is in red, agrees very nicely, not perfectly because it's an approximation, but very nicely with the instantaneous equilibrium, which is dashed. And the old one without the application kind of lags behind it, right? So we've successfully managed to control this and even get the distribution, get the distributions to really, overlap with each other quite nicely. Now this is just a, 2 genotype example. Can we do this numerically in more complicated cases? So let's return back to that 16 genotype example for yeast. And here, if we plot out, so here I just chosen to look at several of these genotypes, so the kind of dominant ones. The dashed lines represent the values of the means of the instantaneous equilibrium distributions. And here is our original protocol. Let's say we're increasing this drug pyrimethamine, and we're using, of course, all the experimental fitness values. So it's in some sense a numerical simulation of what will eventually be hopefully an actual experiment in the labs of our collaborators. And in that original dosage, you see that the actual genotype frequency, which is shown in red, this is one realization of it, lags behind the instantaneous equilibrium values. So we were off target. Now what happens, and of course we can see this, at different snapshots. These graphs essentially represent, the circles represent the sizes of the population, and the pink ones are the actual sizes. The blue ones, or light purple ones, are the equilibrium, and the overlap is kind of shown in this purple color. So you can see here that where you see essentially, discrepancies are places where you have lag between the actual and the equilibrium. Now, if we implement this CD protocol, and again, it's an approximation, so we have to figure out what drug dosage, what perturbation in the drug dosage gives us the best, the closest approximation to that extra term that we need to add. And that extra term, of course, is this complicated multidimensional object, but in the space of all drug dosage, we find the one that best approximates it. And of course, we have to also put in realistic constraints. We can't overdose, the system. So there may be some realistic cutoffs which we've implemented, right? That we're not allowed to go beyond this amount of concentration. Despite all these constraints and despite this approximation, once we find the one that most closely matches our predicted CD term, you see that the solid curves now stay on target really, really nicely. And not just the dominant ones, but also the subdominant genotypes also follow their targets in a really nice way. So, with this perturbation, we're able to essentially get this really nice control of this evolutionary system of many, many different genotypes. And this is for one drug, but we also in the paper, we looked at other drug combinations. We looked at synthetic, we perturbed the drugs, we created kind of fake drugs just to see how this works in generality. And this is not a one-off thing. We generically speaking get this nice, control. It may not be perfect in all cases, but we definitely get a reduction of the lag time in these systems. Now, this is of course just showing the comparison of the mean values. But if you look at the actual, for example, KL divergence between the full distribution and the instantaneous equilibrium, so I call it rho here, but it's P equilibrium. This is the KL divergence. In the original one, the KL divergence spikes up to a large value and eventually goes down as you equilibrate, but with a long lag time. In the CD one, you get a much, your KL divergence is much less, right? And goes down to its minimum value much quicker because you've eliminated that lag. So you really are achieving approximate control of the entire distribution when you do this. So what are the next steps? So this is kind of just the kind of future plans that we're doing. Well, one of the things that is kind of an obvious next step is to take this now and to see this in the laboratory, right? Because everything that I've described right now is a theoretical concept and numerical simulations. But we have collaborators, so Dan Weinreich at Brown and Kerri Geller Samerotte at Arizona State, who have these transgenic yeast in their labs. And we're working with them to basically set up these types of control experiments, actually try to work this out in a laboratory setting. And we of course anticipate, as all, we live in a nice, simple theoretical world. Of course, in real life, when we do this experimentally, there might be other complications. We'll have to maybe extend the theories to deal with that. But we're optimistic that we can maybe translate these ideas and actually see this type of control in an experimental setting, which will be the first time that you can achieve this kind of fine-grained control even as a proof of concept in an evolutionary system. And so that's one plan. We also think that as a general rule, adding more control knobs to the system should increase controllability, right? So if we're piloting a plane, the ability to bank left and right and to move our nose up and down, that helps, right? We have two kind of orthogonal, control directions. And similarly with drugs, if we have multiple drugs which we're applying simultaneously, which do somewhat different things, right, we should improve controllability. So for example, if we need to approximate that added perturbation in our equation, having more drugs at our disposal, more control knobs, should allow us to get a better approximation. So that's one of the things that we're looking at. And this kind of leads to an interesting question of what does it mean for drugs to be orthogonal, right? So, if two drugs do the same thing, right, they're not really useful to put both of them as control knobs. But if they do something very different, it is, but how do we measure that difference? So that's another thing that we're exploring to see, in terms of controllability for these systems. And then the final thing which I wanted to mention is, mathematically, all of these things, I described an evolutionary system, right, where we're talking about, transitions between different genotypes, but there's nothing special about that mathematically, right? So if I were interested in epigenotypes, so we know, for example, that cells have different gene expression patterns, right? And this is actually quite important because, what makes a lung cell different from a liver cell, different from, you know, other types of cells in our bodies, is the pattern of gene expression, the so-called epigenetic, state of the cell. And you know, transitions between different epigenetic states are also really important in things like drug resistance and cancers, right? Oftentimes, it's not a mutation that causes the resistance, it's some pattern in gene expression, even though the underlying genome is the same. And so that dynamics of how you change from one epigenetic state to another, it turns out you can also use that, you can also describe that as a Fokker-Planck, and think of it as in kind of a landscape picture. And in fact, this goes back, you know, more than half a century to, this is known as kind of a Waddington epigenetic landscape. It's, you know, adapted from a famous, description of this by Waddington. And what this is supposed to represent is kind of a landscape where you start with kind of a stem cell at the very top, and kind of like you're biased towards, you know, some kind of differentiated state. And in a way, this is a kind of a stochastic process of diffusing down this landscape and ending up in particular cell types. And what people have begun doing is, of course, we can take the differentiated state, reprogram it back to stem cell state, and then control it and move it into another thing. And there, the ability to control things in finite times, you know, so you want to go from a stem cell to a differentiated cell as quickly as possible and have some degree of control of that. That's going to become really important, right? Because, you know, that's the essence of the control problem there, is to be able to do this in a reasonable amount of time. So we think that these mathematical ideas, one of the, I think perhaps the most exciting future extension would be to developmental biology. But I also mentioned very briefly along the way ecology. For example, we can think of systems of competing species where what's our external intervention? We might imagine like a hunting protocol or a culling protocol. Let's say you're dealing with a fishery and you're looking at a particular part of the ocean, and you have some protocol of how many fish you're going to catch, you know, every season. So you can then change that protocol over time to basically get some kind of desired outcome, to move a distribution of species from one type to another. And it might be to get rid of an invasive species, it might be for some other purpose. But you can imagine some degree, you might want some degree of control over distributions of species. And again, the mathematics of that is very, very similar to the types of mathematics that I've already described in this talk. So we're hoping to also think of these ideas in those other contexts. So with that, I just want to thank all the people involved in this project. So at Case Western, in particular, my graduate student Shamreen Iram, she was the co-first author with another undergraduate co-first author, Josh Cheel, and also contributions from undergrads and grad students, Julia Polesko, Ozench, Gungur Ben-Kuznets-Speck, and then the collaborators in the Cleveland Clinic who first kind of got me started along this whole evolutionary dynamics, thinking about it. Jake Scott, who leads the group there, and then Emily Dolson was his postdoc. She was another co-first author. Nikhil Christian was a graduate student who was working with him, and then my former postdoc Effa Ilker, who worked with me on this, and Sebastian Defner from University of Maryland, Baltimore County, who first introduced me to some of these quantum control ideas. So with that, I'm happy to take further questions and continue the discussion.
Okay, so you have any questions from the audience?
Right, can I ask a question?
Yes, please go ahead.
All right, I say thank you very much for a very nice talk. So I'd like to make the point clear to myself. So what information do you need to determine the counter-diabetic protocol for the selection coefficient S tilde? So do you need to measure the time variation of the mutant concentration for all the time period, or just?
Yes. So in this approximation that we're looking at, which we think is valid for a certain regime of systems, what you essentially need to know is this average x as a function of lambda. So if we know that, then we know how to calculate this perturbation here, because that perturbation just depends on how that average x changes over time. So this is actually, I framed this entirely as like, we want to achieve this instantaneous equilibrium distribution, but you can think of this a little bit more generally as, let's say you want just the system to follow a sequence of distributions. In this particular case, what you need to achieve that is essentially, knowing the mean of those distributions. And this is just a limit where these distributions are somewhat narrow, so that becomes the most important piece of information. But more generically, you would need to know, the full sequence, so P equilibrium, and that you can then use this exact method to calculate it. Yeah, so it's a lot of information.
Right.
And that's going to be one of the major challenges going forward in a practical sense. Now, for these well-controlled laboratory systems where we have these fitness landscapes measured, we think we can do this as a proof of concept. But then the question is, let's say a patient comes in with an infection, right? I'm not going to sit and calculate growth rates as a function of different drugs, for every variant in that infection. So then the question is going to be, can we do this on the fly, right? And there, I think there's some hope, maybe even using some techniques like machine learning, where you begin to kind of give perturbations to the patient and see how they respond, and use, for example, the machine learning model to make a best guess of the underlying landscape. Then once you have a guess of the landscape, calculate the CD protocol, apply it for the next step, see how the system responds, and then based on that response, correct your model and make a better guess, going forward. So in this iterative way, we think we might be able to even do this, even if we don't have the full landscape.
All right, great. Yeah, thank you very much.
Yeah.
Okay, so Professor Beck, Seung-gi, please.
Yes, my question is actually almost the same as the previous one. So the xi bar of lambda in your new solution, I was wondering if it should be obtained from the equilibrium distribution or from the actual distribution.
No, from the equilibrium distribution.
Okay.
Yeah. So the idea is that let's say you have some original protocol where let me do this in this case, where this is kind of like the protocol that describes, okay, I want to get from some initial point to some final point over some amount of time.
Mm-hmm.
Then for each point in that protocol, I can calculate an equilibrium distribution,
Mm-hmm.
and for that equilibrium distribution, I can calculate the average, genotype frequency, and that's essentially shown as these dashed lines. So these dashed lines are basically the X bars. And if I know that, I can then calculate what the perturbation I need to basically make my system follow along the dashed lines. That's the essential idea.
I see. Thank you.
Okay. So any other question, please?
Yeah, can I ask another question?
Go ahead. Yeah.
Yeah, so here in the experiment, they talk about like 16 genes, right? For like distinguishing different genotype.
Mm-hmm.
But I think Plasmodium, the malarial parasite, has like 5,000 genes. So to be like more accurate, the whole genome sequencing needs to be performed and see which genes actually change. So I wonder they have done that type of tests to identify this sequence gene.
Yeah, so that's a wonderful question. And so just to be clear, we're not doing this directly, or I'm not doing anything. I'm a theorist, but the experimenters who did this weren't doing this directly in Plasmodium. They took the gene from Plasmodium and put it in yeast. But regardless of which system you're dealing with, you're totally right that even if I'm focused on one gene, which I think gives me the biggest change in drug resistance and as the biggest contributor to drug resistance, inevitably, if I begin doing this as an experiment, I will accumulate mutations in other parts of the genome. And because there are so many, the other parts of the genome are so much larger than the gene of interest that I'm looking at, I'm much more likely to see mutations there, right? I'm much more likely to see a background mutation, before I see any mutation, among this, at these 4 individual sites. So how does that change? How does that change the problem? So this is something that the experimentalists, we were just discussing this a few weeks ago with Kerri Samerotte, and she's starting to run these experiments. So they have a technique called barcoding, where you can essentially keep track. You create like 500,000 different barcodes, placed in the genome of your cells, so you can essentially keep track of lineages throughout time. And so we can begin to see the accumulation of kind of background mutations, or at least some information about that over time. And what we believe, what this means practically in our model, is that let's say we have a bunch of cells which are all the 1111 genotype in that particular DHFR gene. In reality, because all those cells will somewhat differ in their background mutations, they'll actually have a distribution of fitness values. So rather than each S for each type being basically like a single number, it's going to be a probability distribution of fitnesses. And what that means is that we'll have to generalize this model to deal with essentially, our S of I now becomes a distribution of fitness values. But we believe that that mathematical generalization should not be a problem. But we'll have to, and we can establish those range of fitnesses experimentally as well. So we can take those ones, all the 111s, and just see, what range of kind of fitnesses we will see in that population. But you're right that we do in a real experiment, we do have to think of that as well, all the accumulation of background mutations. So that's one thing. And the other thing that can occur is not just background mutations, but also epigenetic changes, right? So even if the cells have the same genotype, they have slightly different genetic expression levels, and that will be very similar in the sense that timescales of those changes are going to be much faster than timescales of mutations, and you'll get this kind of distribution of fitness values as a result.
I want to ask a question.
Mm-hmm.
One of the last pages of your slides shows about the question about the orthogonal, and could you elaborate more about this orthogonality in the context of your talk?
Yeah. So what I mean by that is, let's imagine that I have a drug, like for example, this drug here, pyrimethamine, which I found that if I increase it in the middle of my protocol, it gives me just the right push to keep my distribution on target. But more generically speaking, sometimes I especially while things are changing quickly, it's hard for me to get, to reach the target. In that case, if these curves diverge, it means I don't have enough control knobs to really precisely travel that path. So I might want to include another drug into the mix. Now, if the second drug that I include in the mix is very similar to pyrimethamine, maybe because on a molecular basis, it does the same thing to the organism, it's not going to give me much of a benefit, because whether I increase that drug or I increase pyrimethamine, they're more or less going to have similar effects. So mathematically, I'm not going to achieve any greater control. But on the other hand, if my second drug does something very, very different from pyrimethamine, right, it affects other different types of genotypes, maybe in different ways, I now have a higher dimensional control over my system. And in principle, that should allow me to reach my target, more precisely. That's the hypothesis. It's something that we're still working on. But I think just as a, intuitive notion, it seems to make sense that we should be able to achieve greater control with more.
So it seems to me that the orthogonality or that kind of concept applies to linear vector spaces, and if your cityscape or landscape is very linear, maybe that could be applied to that globally, but locally, okay, orthogonality might work, but I think orthogonality.
I should mention that orthogonality here, I'm using it in a very loose way, because we don't believe it's going to be just as simple as taking two vectors and taking the inner product of them, right? There might be some complicated function that describes, the differences between the drugs, so we don't necessarily expect it to be just a standard inner product.
Yeah, so as it moves from one point in cityscape to another point, that orthogonality might change into something else, so on.
No, that point is completely taken. So yeah, it's something that this is something that we're working on right now. We're just at the point where we're just adding, one of the first issues is how do you combine multiple drugs together, and there are existing models. People like Yuri Alon, Roy Kishoni have worked extensively on this, of like, how if you know the response of an organism to one drug and another drug, and you now combine the drugs together, can you predict what the combination does? So we're building on those types of models as well, and those are highly nonlinear models typically. So there's a bunch of intermediate steps to seeing how multiple drugs work together.
Thank you so much for this interesting talk.
Okay, thank you.
Mike, I have one question. So your whole goal is to improve the controllability of this, whatever the fitness landscape of some bacteria or maybe some disease-causing agents, and supposing I have like a maybe multiple orthogonal drug, like 10 multiple orthogonal drug, and then if you have like a high controllability, then what's your ultimate goal? Like you can get rid of the drug resistance from your system?
Yeah, I mean, if your drugs, of course, are effective and you can just kill them, you know, that's all. In the end, you don't need control if you can kill it, right? So if you can just nuke the system, you're fine. But if you're in a scenario where applying any simple application of drugs leads to resistance, you might want to play these tricks where, like, the goal of this initial control is to actually get the population into one genotype, into one type, right? So you in some sense, you want to, your patient may come in with many different types of genome, a mixed distribution, and you're going to apply this initial, control step to get them all to be one particular genetic variant. Because if you then have a drug which is works super well in that particular genetic variant, you can then wipe the population out. So that's the idea. Is maybe you're going to do this as a first step of like a multi-drug, multi-stage kind of protocol. So that would be kind of the evolutionary medicine version of this.
So this is something related to some clinical strategy.
Yes.
You administer some drug A and drug B. The order is also matters, right?
The order matters a lot, and this is, I mean, just practically speaking. I mean, this is beyond. I mean, I'm not a doctor, but my collaborator is an MD as well, and what he says is now, for example, let's say you go to the doctor and you have a very bad infection. And they'll prescribe to you an antibiotic, and they'll tell you, okay, let's see what happens. Come back to me in 2 weeks. You come back in 2 weeks, and let's say the infection is not getting any better. They're going to just look at their computer screens. Out of the 8 different antibiotics that they're allowed to prescribe, they're going to just randomly choose a second one, and they're going to continue this process. But there's no rational choice. The first one that they gave you, maybe they liked it a lot. Maybe it seemed to work with lots of patients. They kind of are basing this kind of on their anecdotal evidence. And the second one, they're also going to say, oh, well, it didn't work for you. The first one didn't work. Let me just choose another random one. And there's no rational thought behind which order in which you do this. But we know from the evolutionary perspective that, applying drug A and then B can lead to very, very different results than B first and then A, right? So the whole idea of this thing is if we understand something about the underlying evolution, we can design rational drug protocols to help fight infection better. Yeah.
Okay, thank you. So we have some more time. You guys have any other questions, please?
Can I ask something?
Yeah, sure, please.
Yeah, thank you for the nice talk. I thought this was really cool. Is there a way to apply this kind of framework to think about minimizing the time or the total dosage of the drug?
Yes. So that's another aspect that we want to connect to in the future, because right now we focused on, like, how do we follow a target path, right? And that was and it turns out that it's nice because you can then have these close form analytical solutions, but there's a broader field of like optimal control theory where there's some kind of loss function which you want to minimize, and that might be a mixture. You might choose your loss function to prioritize time and dosage, right? There might be other, in generic, like cancer therapy, like maybe you want to minimize the amount of chemotherapy you give, stuff like that, because it has all these bad side effects. We believe this CD, this counterdiabatic is one special case of kind of a general optimal control protocols. And one of the things we plan to do is kind of, see how that connection works out, because there have been people who worked on optimal control theory versions. Oftentimes you end up with kind of very complicated protocols, which you have to implement, you can only solve numerically. This has some nice advantages in that it's kind of this close form analytical solution, but we want to connect the two. We want to see how they relate to each other. And so hopefully in the future, we can broaden this and generalize it to other types of, control problems, not just following a target path.
Thank you.
So Mike, this evolutionary relaxation time, if you actually change the parameter for lambda 1 to lambda 2, there's some evolutionary relaxation time, right? If this relaxation time is very, very slow, then your protocol, CD protocol still works, or what's the limitation?
It works. It works, but you then have to, that means that the lag is very large. Mm-hmm. So generally speaking, the larger the lag, the stronger you have to, nudge it. So basically you need to apply more drug, right? So there's going to be some limits in the sense of like, you can't overdose, you know, the, you know, you can't apply like an infinite, you know, delta function of drug to make things go infinitely fast, right? So there's going to be some limitations. For the systems that we were modeling numerically, we can work within kind of like constraints, right? Put in some maximum drug dosage, it's still worked out. But that's just because here the mutation times were not, you know, so extreme. So there might be limitations in that as well. If the mutation times are just too insanely slow, there might not be able to have the freedom to speed it up. Now, we may not be able to speed it up to make completely reach the target, but maybe if we get halfway there, right, that's still a benefit. Right, so there's, you know, speeding things up even partially is, you know, with an approximate protocol still has some uses. Yeah, but you're right that the timescales will matter a lot.
So, okay, so any other questions? No, then, okay, let's thank this exciting talk to Mike. Okay, so let's thank. How do I, like there's a, okay, so Mike, thank you very much.
And thanks again for inviting me, and yeah, and for all the great questions.
Yeah, I enjoyed your talk a lot. Okay, so I think we will be contacted by Miss Kim again.
Okay.
Yeah, regarding what. Okay, thanks.
All right. Yep.
Yep.
Right. Have a good morning.
Yeah. Have a good evening.
Yeah.
Good night.
Yep. See you.
Yeah.